Assalamualaikum everyone. Today we're going to talk about the periodontal ligament. The periodontal ligament is a part of the periodontium, which is the supporting tissue and the investing layer of your tooth. It consists of two hard tissues, cementum and the bone lining the alveolus. Between these two tissues, there is a soft connective tissue, the periodontal ligament, that is attaching to cementum on one side and the bone lining the alveolus on the other side. The fourth part of the periodontium is the gingiva facing your tooth. So here in this figure, this is your root part of the tooth. This basically is your outermost layer of the root, which is your cementum, and this hard tissue here is your bone lining the socket, the alveolus. And between these two, this whole soft connective tissue is the periodontal ligament. So this periodontal ligament basically helps the tooth be embedded within the bony socket and sustain its position there. Periodontal ligament basically, unlike the other hard connective tissues of your tooth, is a soft specialized connective tissue situated between the cementum and the bone lining your socket. So here you have the cementum, the outermost layer of your root, and here you have the bone lining the socket, and between these two, this whole soft connective tissue space is the periodontal ligament. It consists of certain fibrous ligaments and fiber bundles that connect to the alveolar bone on one side and cementum on the other side, as you can see, and they're attaching your tooth and embedding your tooth within the tooth socket. The width of the PDL space varies from about 1.5 mm to 0.38 mm, and it is thinnest at the mid-root region, somewhere here. The width of the PDL also decreases with age. In this figure, you can clearly see the bone on one side and the PDL on the other side, root on the other side, basically the cementum. And between the cementum and your bone, this soft connective tissue space that you can see is the periodontal ligament. In this figure, you can clearly appreciate the difference in the let me change the color of my highlighter. It's not very prominent. Okay, so this figure clearly shows that the width of the PDL space is changing. At this point, it is the widest, and at this point, it is the thinnest. So this would be the mid-root region probably. So the width of the PDL space varies along the root length, and it also changes with age. With age, the PDL space becomes thin. In this figure, you can see the dentine lined by cementum on one side and the bone lining the alveolus on the other side, and between these two you can see the ligament space, which is filled with tissues and fibers. The fibers of the periodontal ligament extend and embed into cementum on one side and into the bone lining the socket on the other side. And this embedded part of these fibers, this embedded part of the fiber within the cementum and the bone, is called the Sharpie's fibers. So the Sharpie's fibers basically are the embedded portion of the collagen fibers of the periodontal ligament that are embedded in the bone on one side and into the cementum on the opposite side. And these Sharpie's fibers are basically embedding the tooth within the bony socket. So this is how the PDL space is helping the tooth sustain its position, and it is helping the tooth being embedded within the tooth socket without falling off or coming out. So by composition, PDL space does not have any mineral. It only has soft connective tissue, which consists of extracellular matrix comprising of the ground substance, the non-collagenous matrix proteins, and certain fibers. The non-collagenous proteins consist of GAGs, proteoglycans, glycoproteins, glycolipids, fibronectin, tenacin, and the fibers consist of collagen fibers type 1, 3, 5, 4, 6, 8, and 12, along with oxytocin fibers, reticulin fibers, elastin fibers, and the cells embedded within this extracellular matrix are synthetic type, the fibroblast, cementoblast, osteoblast; resorptive type, the osteoclast, the cementoclast, and the fibroclast; certain defense cells, epithelial cell rests of Malaces, and undifferentiated mesenchymal cells. So you can clearly see that there is no mineral component. There is ground substance into which fibers and the cell communities are embedded. So we are going to individually talk about all the components of the periodontal ligament, starting with the cellular community. The cells of the PDL are basically divided into different cell types according to their functions or origin, basically. So the synthetic cells are the cells that obviously produce certain tissues or something. Then the resorptive cells are the ones that resorb the tissues. Progenitor cells are the ones that could be source of new cells. Then we have the epithelial cells, which are epithelial in origin, and then we have certain cells which have defensive functions, so we classify them as defense cells. So the synthetic cells that the PDL has are the fibroblasts that synthesize collagen, osteoblasts that synthesize alveolar bone, cementoblasts that synthesize cementum. Then we have the resorptive cells, which consist of fibroclasts that resorb or degrade collagen, osteoclasts that also degrade bone, and cementoclasts that resorb the cementum. Then we have undifferentiated ectomesenchymal cells, or the stem cells, that could produce or that could differentiate into any of the fibroblast, cementoblast, osteoblast, depending upon the stimulus. They could be source of any of these cells. Then we have the undifferentiated, not the undifferentiated, we've talked about that. We have the epithelial cell rests of Malaces, which are actually the remnants of the Hertwig epithelial root sheath, and they are present within the PDL space, where they help in the repair and the regeneration of the PDL space. Then we have mast cells, macrophages, and eosinophils that have defensive function, so we classify them as the defense cells. Now we are going to individually talk about all of these cells, how they look like, where they are present, and what is their function within the PDL space. Starting with the synthetic cells, the most abundant cells within the PDL are the fibroblasts. Fibroblasts basically originate from the ectomesenchyme of the dental follicle. These cells basically have a flattened disc shape, or they could be fusiform with numerous fine cytoplasmic extensions. They have a centrally placed nucleus, and they have abundant amount of protein synthesizing organelles and a very well developed cytoskeleton, as well as an extensive actin network. These cells secrete and synthesize proteins, and they degrade collagen and produce collagen as well. They have an extensive actin network which helps them change their shape and migrate from one part of the periodontal ligament to another part. So movement and migration is possible for these cells because of their actin network, which indicates the functional demands of the tissue. Here you can see the cells, fibroblasts basically. These are disc-shaped cells with a centrally placed nucleus, and you can see the numerous cytoplasmic extensions that they have. This cell has quite many cytoplasmic extension, and here they look like fusiform. So they could be either fusiform with a centrally placed plump nucleus and disc shape with many cytoplasmic processes. These cells basically have an exceptionally high turnover of the proteins because they have the ability to synthesize collagen and degrade it as well. So because of this ability, they have a high turnover. These cells are connected to one another within the PDL space through gap junctions and adhering junctions. They are regularly distributed throughout the ligament, and they are oriented parallel to the long axis of the collagen fibrils of the PDL, and the processes of the fibroblasts, they wrap themselves around the bundles of the collagen fibers. They are the fibroblasts of the PDL, basically they generate an organizational pattern, and they have the ability to both synthesize the extracellular matrix protein and shape it, into which the collagen fibril bundles are inserted. The collagen fiber bundles of the PDL, which are inserted into bone and tooth on one side, the Sharpie's fibers, these embed within the extracellular matrix. And that extracellular matrix basically is both synthesized and shaped by these PDL fibroblasts. The fibroblasts basically have a very important role during post-eruptive tooth movements, including mastication, accommodation of the jaw growth, compensation of the occlusal and proximal wear, and compensation for the mesial drift. Proximal wear is basically when two adjacent teeth, when they come into when they contact at the proximal surfaces, wear occurs between those proximal surfaces. So that is called the proximal wear. And similarly, mesial drift is when the adjacent tooth is removed for some reason, and the tooth adjacent to that space tries to move into that space. That is called the mesial drift. The tooth mesial to it when it tries to move into that newly created space. That is called the mesial drift. So the fibroblasts basically play a very important role during all of these movements. And how does it do that? Whenever the stress is placed on the tooth in the form of mastication or occlusal wear or proximal wear, that stress is distributed from the tooth into the periodontal ligament, and that mechanical stress is perceived by the fibroblasts as a stimulus. The fibroblasts, in response to that mechanical stress, produce the extracellular matrix. That is how they sustain the periodontal ligament during function. If there is any alteration in the fibroblast function, the PDL tissue would be lost because the collagen would not be produced, and as a result of reduced production of collagen, the PDL tissue would be lost. Fibroblasts at the same time secrete certain proteinases called the matrix metalloproteinases, which degrade its own matrix, and they also produce the tissue inhibitors of those matrix metalloproteinases, and these tissue inhibitors are present in high amount in the healthy PDL, which do not let the degradation of the collagen and in turn PDL in the healthy tissue. Whereas in case of certain diseases like inflammation and any other periodontal disease, the expression of the matrix metalloproteinases, the MMPs, is increased, which leads to aggressive loss of collagen within the PDL at that particular disease location and eventual tissue destruction. So the fibroblasts, which are predominantly present within the PDL, have these important functions of generating the extracellular matrix and the fibers of the PDL, and also maintaining them and degrading them when required. So the fibroblasts have disc-shaped bodies with a centrally placed nucleus, active extensive actin network, protein synthesizing organelles. They secrete and synthesize proteins. They can move around the the PDL space. They synthesize collagen, and they can also degrade the collagen that they produce. The next type of the synthesizing cells within the PDL are the cementoblasts. As the name says, the cementoblasts are the cementum forming cells, and they line the surface of the cementum. These cells are basically squat cuboidal cells with a large vesicular nucleus and one or more nuclei and abundant cytoplasm consisting of all the cytoplasmic organelles that are required for protein synthesis, because obviously actively secreting cell has loads of protein synthesizing organelles because it has to secrete proteins. So these actively secreting cells, the cementoblasts, are actively depositing cellular cementum, and they exhibit abundant amount of basophilic cytoplasm and cytoplasmic processes. As you can see here in this figure, this is your periodontal space, and this that I'm highlighting, this region that I'm highlighting, is basically your cementum, and these black cuboidal cells that you can see here on the surface of the cementum are the cementoblasts. These are cuboidal cells, they're actively secreting cementum and they're lining the surface of the cementum. On the opposite side of the PDL, the bone side of the PDL, where you have the alveolar bone, on the surface of the alveolar bone, or on the surface of the bone lining the socket, you have these colony of cells which are the osteoblasts. So we have the osteoblasts lining the bone on one side, the cementoblasts lining the cementum on the other side, and in between we have the PDL where the abundant amount of fibroblasts are organized. So the osteoblasts basically, as I've said, are the bone forming cells, and they're lining the tooth socket. They very closely resemble cementoblasts. The osteoblasts layer, however, is only prominent when the bone is actively forming. However, at all other times when the bone is not actively forming, these cells are flattened and they're inactive, lining the bone cells, and we call them the bone lining cells. However, the active osteoblasts are cuboidal cells. They have a prominent round nucleus at the basal end. They have lots of protein synthesizing organelles, the rough endoplasmic reticulum, mitochondria, and vesicles. And these cells, they appear to be basophilic due to high amount of rough endoplasmic reticulum, and they're connected to one another through desmosomes and tight junctions. So this was all about the synthesizing cells, where we have these osteoblasts that synthesize the bone, the cementoblasts that synthesize the cementum and they're lining the cementum, and then we have the fibroblasts that are actively secreting the collagen and the matrix of the PDL. Then we have certain resorptive cells within the PDL, and as the name says, these cells, they resorb. Osteoclasts would resorb bone, fibroblasts would resorb collagen, and cementoclasts would resorb cementum. So starting with the osteoclast, they resorb the bone and they arise from blood cells of the macrophage type. These cells, they have a wide variety of shape and size, ranging from large multinucleated cells to small mononuclear cells. And when an osteoclast has to resorb the bone, specifically alveolar bone in this case, that particular surface of the alveolar bone produces a concavity or a depression which is called the resorptive lacunae or the Hauschip's lacunae, into which the osteoclast resides, or it gets stuck in there. The part of the osteoclast that lies adjacent to the bone has a striated appearance, and that part of the cell, that part of the cell membrane throws itself into a myriad of deep folds, and that border of the osteoclast is called the ruffle border. The cytoplasm of the osteoclast right inside the ruffle border does not have any organelles, and it is called the clear zone. However, this zone does have certain vesicles that contains acid phosphatases that help in resorption. Any resorption that has to occurs through the ruffled border. The osteoclast attaches to the bone surface that it has to resorb through the sealing zone. The sealing zone basically is attaching your osteoclast to the bone tightly, creating a compartment or sealing the compartment where the resorption could occur. Between the sealing zone and the bone surface, there is a layer of interfacial matrix called the lamina limitans. So this is how the osteoclast would look like. This is how the osteoclast is shaped. Let me open up the pointer. This is how the osteoclast is shaped. You have a functional secretory domain, then you have the basolateral membranes, and you can see multiple nuclei because it is a multinucleated cell. The bone that it has to resorb has made a depression or a concavity. This is called the Hauschip's lacunae or the resorptive lacunae, into which the ruffle border is fixed. The ruffle border, or into which the osteoclast is basically fixed. That part of the osteoclast, this part basically, that is closely attached to the bone, or that is near the bone that it has to resorb, it looks striated. As you can see, it has striated appearance, and this striated appearance is because the osteoclast membrane or plasma membrane has thrown itself into these numerous folds. This border of the this whole border of the osteoclast is called the ruffle border. All the resorption is occurring at this face of the osteoclast. इस फेस के अगेंस्ट कुछ नहीं हो रहा होता, सिर्फ इस फेस के अगेंस्ट, रफल बॉर्डर के अगेंस्ट रिजॉर्प्शन हो रही होती है, बिकॉज इट इज क्लोजली अटैचिंग टू दी बोन surface. The part of the osteoclast that is attaching or helping it attach to the bone surface is this, the sealing zone on one side and the sealing zone on the other side. ये tightly एक ऐसा environment create कर लेता है जिसकी वजह से ruffle border seclude हो जाता है external environment से. Between the sealing zone and the bone, there is a layer of interfacial matrix present. इनके दरमियान में एक layer होती है lamina limitans की. However, the ruffle border, the cytoplasm of this is the cytoplasm of this basically everything inside the cell is the cytoplasm of your osteoclast. The cytoplasm right inside the ruffle border, this cytoplasm basically does not have any organelles, and it is called the clear zone. So anything below this region basically is the clear zone, and I'm labeling it as CZ. This does not have any organelles. However, this part does have certain vesicles which are filled with acid phosphatases. All of the resorption is occurring at the ruffle border. Nothing is occurring at the functional secretory domain. This is all you need to know about the osteoclast. The osteoclast is a multinucleated cell. It could be a small mononuclear cell. The resorptive, the bone that it has to resorb, develops a concavity called the Hauschip's lacunae, into which the osteoclast resides. The part of the cell that lies adjacent to the bone has a striated appearance because of the membrane throwing itself into myriad of deep folds, forming a ruffled border. At the periphery of this border, the plasma membrane is closely opposed to the bone surface, and the adjacent cytoplasm does not have any organelles, so it is called the clear zone. However, it does have abundant vesicles, the acid phosphatases in the cytoplasm, which help in resorption. Attachment occurs via sealing zone, and the resorptive activities occurring at the ruffle border. There is an electron-dense interfacial matrix layer called the lamina limitans present between the sealing zone and the calcified tissue. So that is how your osteoclast would look like, and for now you need to know if it is a multinucleated cell, what is the shape, how does it attach, what helps in the attachment, where the resorption is occurring. That's it. We're going to go into the details of how the resorption occurs and the steps of resorption when we're going to study the bone, inshallah. The next resorptive cells are the fibroblasts. These cells rapidly degenerate the collagen that they produce by phagocytosis, and they form the basis of the rapid turnover of the periodontal ligament because they provide the machinery for degradation of collagen as well. The extracellular element that helps in the degradation of collagen includes the collagenases, which basically cleave the triple helix assembly of the collagen molecules within the fibrils. Cementoblasts actually are the cells that resorb the cementum. Cementum is not remodeled in the fashion as the alveolar bone and the periodontal ligament is remodeled. These cells resemble the osteoclasts and they are found in normally functioning periodontal ligament. Cementum, however, is not as often remodeled as does the alveolar bone and the periodontal ligament. Their origin is unknown, but they're believed to originate to share their origin with the osteoclasts. So this is all about the resorptive cells. So we had the osteoclast resorbing the bone, fibroclast degrading the collagen, and cementoclast resorbing the cementum. Then we have the epithelial cells. The epithelial cells present within the periodontal ligament are the epithelial rest cells of Malaces, which were the remnants of Hertwig epithelial root sheath. They lie close to the cementum at about 25 micrometers from the surface, and these cells are basically clusters of epithelial cells or strands of epithelial cells surrounded by basal lamina, and these epithelial cells are basically cuboidal cells with a deeply staining nucleus. So this is how they would look like. You would have clusters of cuboidal cells with a deeply staining nucleus surrounded by the basal lamina. एक्सटेंड हो गया बेसल लेमिना सराउंडेड. सो यू वुड हैव अ बेसल लेमिना कवरिंग जिसके अंदर आपके पास न्यूमरस epithelial cells होंगे cuboidal shaped. These epithelial cells or these rest cells of Malaces, they help in the repair and the regeneration of the PDL, and this is how you would see the epithelial rest cells under the microscope within the PDL space. The next type of cells that we have within the PDL space are the progenitor cells, which are the undifferentiated ectomesenchymal cells or the stem cells. These cells are basically source of new cells for the PDL, and they're present along the blood vessels within the PDL, and we say that they have a perivascular around the vessel's location. These undifferentiated ectomesenchymal cells, as the name says, could differentiate into any type of cell, fibroblast, osteoblast or cementoblast. So we could have a single progenitor cell that could give rise to any type of daughter cell which may differentiate into fibroblast or an osteoblast or a cementoblast depending upon the stimulus. Or there could be separate progenitors for each of these cell lines. For example, there could be a separate progenitor for the fibroblast, a separate progenitor for the osteoblast, and a separate progenitor for the cementoblast. So whenever a type of cell is needed within the PDL space, these undifferentiated ectomesenchymal cells receive that stimulus and they differentiate into that particular cell type which is required. So they are basically a source of new cells within the PDL. There is also selective deletion of the cells of the ligament happening by apoptosis within the periodontal ligament as well to maintain the balance between newly forming cells and the old cells. The next type of cells that we have within the PDL space is the defense cells, and we're going to talk about the defense cells in the next lecture. And in the next lecture, we're also going to talk about the fiber system of the periodontal ligament. The attendance and the question and answer for this session is going to be at 1:30 Wednesday, 1:30, 10th of June, inshallah. And I'm going to see you guys in the next lecture. Allah Hafiz.